Alrighty. Good morning or good afternoon, depending on where you are across the country. Welcome to our newest edition of our webinar series for the Glycan Trainee Association. My name is Katrina Pegg, and I'm a student at WLU, and I will be the chair of today's webinar. Moving straight forward, it is my great honor to introduce our next speaker for this webinar series, which is Dr. Duong Bui. She obtained her bachelor degree from Belgorod State National Research University in Russia, following which she completed her master's training through the Erasmus Excellence in Analytical Chemistry program at the University of Tartu in Estonia and Uppsala University in Sweden. Dr. Bui then received her PhD in chemistry from the University of Alberta in December of 2023 and is currently working as a postdoctoral researcher at the Klassen and Mahal groups. Dr. Bui is scheduled to begin her appointment as an assistant professor at the University of Waterloo in March of 2026, where she has a lab that focuses on advancing native mass spectrometry, glycoproteomics, and biophysical approaches to elucidate how glycans regulate the immune signaling and how these processes contribute to disease. So, Dr. Bui, if you'd like to take it away.
Yeah. Thanks so much for the introduction, Katrina, and thanks so much for the invitation to talk here today. So in this presentation, I'm going to show a part of my PhD and postdoc research, where I developed and applied native mass spectrometry methods to study protein-glycan interactions to crack the Glycan code. We here, most of us know that our cells are sugar-coated. And in fact, the cells is coated with a dense layer of carbohydrate-containing structure, which is collectively called the glycocalyx. And in fact, glycans are the third most abundant component of the cell membranes after the lipid and proteins. And because glycan coat our cells, they involved in many cellular processes, for example, cell signaling, cell-cell interactions, cell protections. Pathogens such as virus or bacteria also employed glycans as a receptor or co-receptor to attack and infect our cells. On the other hand, our immune cells use a receptor to recognize glycans to distinguish pathogens from our normal cells. And glycans are important in disease. So many glycans are studied to be the biomarkers of disease, such as in cancer, glycan involved in cancer cell proliferations, cancer metastasis. So in our lab, we interested in studying protein glycan interaction or receptor glycan interactions. And to study these interactions, there's different approaches. From my point of view, one of the most effective approaches to use native mass spectrometry. So most of us here know mass spectrometry. So mass spectrometry is analytical technique that we detect the ions to know the molecular weight, or most of the time, actually, we know the mass to charge ratio. So native mass spectrometry is a subfield of mass spectrometry where we mostly use electron spray ionization to ionize ion. And the advantages of mass spectrometry is that not only we can detect the molecular or the biomolecules to know the molecular weight, we can also preserve the non-covalent complex. And by doing so, we can discover the interactions. We can say about their stoichiometry, and we can also study the thermodynamic and kinetic parameters of the interactions. So it's not like the process is quite complicated. However, going from solutions to spectra, there is ionization process, there is transfer of the ions and detection of the ions. However, all of the process are very quick. So what we can imagine is the spectra is actually a snapshot of what in the solution. So in case of native mass spectrometry, instead of using the very big probe, we use a small capillaries as shown here. And if you can see me, there is how small the capillaries is. And for native mass spectrometry, we usually load to 1 to 10 microliter of the sample in the capillary, and which is quite small, with the outer diameter of around 1 micron. So if you want to study the binding of biomolecule, for example, protein-glycan interaction, we want to know how strong they bind together. So we calculate the Ka or Kd or dissociation constant. To do so, we usually extract the abundance of the proteins and the complex. And from the abundance, we take the ratio, calculate the Kd. Advantages of native mass spectrometry are several folds. So as I mentioned, we use low sample consumption. And for native mass spectrometry, protein and glycans are used as they are, so no modification of protein nor glycans. There are no washing step, and it's a fully quantitative technique. So there is different method in native mass spectrometry for different application, different biomolecular interactions. So broadly, we can divide the interaction into two case scenarios. Small ligand and big ligand in comparison to the size of the protein. So when we talk about small ligand, we talk about less than 5% of the molecular weight of the protein. So as I already showed you, if we have a small clean protein and small ligand, we mix them together. We have the abundance, take the ratio of the abundance, and we can calculate the Kd. So the method was already established in 2002 in the Glycan group, and what we call is a direct binding method. However, proteins are not always homogeneous and nice. So for example, the spike protein of SARS-CoV-2 is a glycosylated protein. So usually we see the protein in the simplified version where we have no glycans. In fact, spike protein is heavily glycosylated. So instead of seeing one nice spectrum, we have a broad spectrum that is really hard or impossible to distinguish free protein from the complex. And if we cannot distinguish the complex from the free protein, we cannot take the ratio and calculate the Kd. So how do we study the interaction in this case? To solve this challenge, in 2012, the Glycan group developed a method called the catch and release NMS approach, which is also abbreviated as a core NMS. So in this approach, the protein of interest is used as a bait to mix with a mixture or libraries of glycans. And we spray them, send them to the instrument. Here we can isolate according to the MOVZ, mass to charge ratio or molecular weight. We can isolate the complex from the free glycan. And then we send the complex to the HCD cell where we can dissociate the glycans from the complex. And by doing so, we can detect the free protein and the free glycan. And if we use the glycans in the libraries at equimolar concentrations, what we can do is we can say about the relative affinity or affinity ranking. So imagine what we detect is everything in the complex. Though the stronger the affinity, the more glycan as in the complex. That's why more glycans are detected. So the simplified version of this core NMS or catch and release NMS approach is we can imagine that using the instrument as a fishing rod and the protein as a bait, and then we can fish out the interacting glycans from the pool of other non-interacting ligands. And then after that, we release the glycan from the bait to detect the glycan. And because the glycan are small, when we release them, we can easily distinguish different glycans that bound to the protein and use the abundance to say about the affinity of the glycan. So using this approach in 2022, we applied this to identify the co-receptor of SARS-CoV-2, and it was found that glycolipid, so here GM1, is a co-receptor of SARS-CoV-2 in addition to ACE2. However, at that point, even with more than 100 glycans, we didn't have several structure like many structures. For example, we didn't have access to different type of N glycan or O glycan. So we couldn't examine the interaction of those glycans to the SARS-CoV-2. So that is a challenge in studying protein glycan interactions. So we have the glycan is diverse. So it is estimated that there are more than 10,000 structure of glycan in nature, using like with different monosaccharide linkages and branching. However, the number and amount of purified glycans to study is only a small fraction of that. To overcome this challenge, shotgun glycomics were developed. In shotgun glycomics, instead of having glycan synthesized and purified, glycans are extracted from natural source, for example, from cells and tissues to increase the diversity of accessible glycans. And as a result, from those natural sources, we have focused glycan libraries. And because they are in the libraries or in the mixture, usually it's really challenging to separate each individual glycans, and they are usually at unknown concentration. So how to study or what method to use to study interaction of protein with a mixture of glycan, and then we can quantify the interactions. We use native mass spectrometry. So to study this, in 2023, I developed a method called the concentration-independent mass spectrometry. The basic idea of CoNMS is a slow mixing in the capillary. So imagine in a small capillary, if we load two solutions with different composition, and because of the difference in the composition, there will be this mixing process that create a gradient of concentration of different components. The mechanism behind the mixing mode was studied recently in our group, and Sima, our undergrad student, developed an on-bench setup where she used the iPhone camera to record the changes of the solution with time. And by using mathematical equations, we could deconvolute the factor driving the slow mixing into diffusion, advection, or differential electrophoretic mobility. So go back to CoNMS. So the basic idea is like if we have the protein, the target protein, we can load the protein in two solutions, solution 1 and solution 2, with the same concentration. And the ligand of interest of unknown concentration is loaded in solution 2. And because there's a difference in the concentration of ligand in solution 2 and solution 1, what we expect to see is a mixing of ligand. And with time, we'll see the gradient of ligand concentration. And once the ligand concentration increase, we expect to see the shift in the formation of more complex. So we see that protein is being consumed and complex is being formed. Although we don't know the concentration of ligand, we cannot calculate the KD. However, if we can model the change, we can also estimate the KD. So by doing that, we can quantify the interaction without knowing the concentration of ligand and even mixture of ligand. So using this approach with the libraries of glycan and glycopeptides from natural source, we could study the specificity of different lectin, So here is again example of the receptor binding domain of the SARS-CoV-2. As I showed in the previous slides, that GM1 was identified as the co-receptor of the SARS-CoV-2. And if you can see here, the interaction of a GM1 oligosaccharide with RBDs are comparable with other N-glycan that we didn't have the access before. So in addition to ganglioside or GM1 or glycolipid and glycan could be co-receptor of SARS-CoV-2 as well. So this was confirmed by pseudoviral infection assay performed in the Macaulay lab. So the idea is that the Macaulay lab used kyphonensin as an inhibitor of complex glycan formation. So kyphonensin blocks the biosynthetic pathway of complex and glycan. What we have is a mannoses, oligomannoses, instead of a silated complex and glycan. By doing so, we saw that the infection was decreased. This was further confirmed by the authentic infection assay performed in the Hoffman lab. And also with this method, we also study other lectin, for example, one of the cyclic, which is a cyclic 7 here. So cyclic 7 was found to play important role in cancer progression and immunity. Other studies have found that the binder for cyclic 7 is a GD3 with the reported KD values of around 98 micromolar. And using the COI NMS approach, we found the KD of 97 micromolar, which is very close to the in great agreement with the reported value. And we also found that in addition to some ganglioside and milk oligosaccharide, silated N-glycan are also the binder of the cyclic 7. So if we look at what have found in the literature, we see that the missing piece here is O-glycan. So even with the extracted glycan from natural source, we didn't have access to O-glycan because we then no universal enzyme to release O-glycan. So to overcome that, I use glycopeptides instead. So by extracting glycopeptides from purified glycoproteins of known sequence, we could study the interaction of all glycopeptides with cyclic 7. So we can add the values here. You can see that some of the old glycopeptides have quite comparable affinity or KD values as the reported ligand GD3 and other ligand. And here we can see that the advantages of using native mass spectrometry or COI-NMS is more entries can be added without repeating the measurement. So because we have the fully quantitative value, we can run more experiments later and then add more value to the table. And now we are in progress of extending the libraries to use so for the libraries of glycans from different natural sources. So here is the 2 different batch of glycan libraries from cancer cell, and we are applying the COI-NMS to screen and to identify the interaction of relevant protein glycan interaction in immunity. So that was about the first scenario where the glycans are small. How about when the glycans are bigger than 5% of the protein? I think most of the time the glycan is bigger than 5% of the protein. So in case of big glycan, if we look at the native mass spectrometry process or the electrospray ionization process, we see that there is many things happens. So from solution to the signal that we detect, there is ionization process, ion transfer, and ion detection. And because different molecules have different properties, so they are ionized differently, they are transferred differently, and they are detected differently. That lead to what we call the non-uniform response factor, which means if you have equal concentration of molecules in the solution, we don't have the same abundance detected. So here is example of how that affect the quantification of interaction. For example, if we have protein ligand to form the protein ligand complex, in one set of the parameters, we detect signal that we have ligand, we have protein, and then we have a complex. And using the abundance, we have the KDs of around 37 micromolar. However, with the same solution, if we change the parameters of the experiment of this instrument, we got the KD of 0.1 micromolar. So we see that it's 2 order of magnitude difference. So that's how unreliable it is with non-uniform response factor to calculate, to quantify the interaction. So what is the correct value? Actually, there's no correct values in here. If we compare those values with what was obtained by using ITC, we see that the KD is around 0.7 micromolar, which is not 0.1 or 37 micromolar. So that is a long-standing challenge in native mass spectrometry to correct for the non-uniform response factor to quantify biomolecular interactions. So to overcome this challenge, in 2022, I developed a method called slow mixing mode NMS or slow-mo NMS. The basic idea of slow-mo NMS is quite similar to what I've showed you with coil NMS. Is we also load the tip with two solutions. So we have protein and ligand of known concentration in solution 1. So we have the abundance ratio. In solution 2, we have the same concentration of protein, but larger concentration of ligand. So what we see is we have initial equilibrium where we have the ratio of the signal of the complex and the free protein. And when mixing starts, we see the change in the signal again. So the protein signal is decreasing and the complex signal is increasing. And because it's one-to-one interaction, what we expected, the change is one-to-one. However, because of the non-uniform response factor, we see that the change is not one-to-one. And if we take the ratio of the change, we can have the relative response factor. Using the abundance ratio from the initial equilibrium and the relative response factor, we can calculate the KD. So using the slow-mo NMS for the same interaction, we got the KD of 0.5 micromolar, which is in excellent agreement with the reference value. And notably, these values is insensitive to the instrument or parameter change. So even when we change the parameters of the instrument, the value stays the same. Slow-mo NMS was applied to study like a wide range of interactions. So we have small molecule interaction, we have protein interaction, protein glycoprotein interaction, protein with nanobody in the present or absent of glycan interaction, also the membrane mimetic interaction with the protein. So back to the SARS-CoV-2 again. So we already identified the co-receptor, glycan co-receptor of the SARS-CoV-2 to be glycolipid and N-glycans. The main receptor of the SARS-CoV-2 is ACE2. So when we study this interaction, we have ACE2 interacting with the spike protein of the SARS-CoV-2. ACE2 is a dimeric glycosylated protein. Spike protein is actually a trimeric protein. So each monomer of the spike protein has 22 N-glycosylation site and 17 O-glycosylation site. So it's heavily glycosylated. So that's why we saw a broad spectrum. and ACE2 monomer have 7 N-glycosylation site. And when we mix them together, we have signal of some complex. However, it's really hard to identify what are the complex and where are the complex. So in mass spectrometry, there is a technique called charge detection mass spectrometry, and recently Thermo Fisher developed a technology called the direct mass technology. So in addition to detecting the mass to charge ratio, using charge detection mass spectrometry, we can also detect the charge of the ion. So combining the charge with mass to charge ratio, we can easily convert it to the mass. So here is a deconvoluted spectrum from what we see here. So we see ACE2, we see the spike protein. We also see one-to-one complex, the dimer of the spike protein, and then 2-to-1 complex. And then using the charge detected here, we can estimate the location of the different complexes. So we have one-to-one complex in this region. This region have both dimer of the spike protein and 2-to-1 complex. And by detecting these species, we can propose like different pathway of complex formation. So from spike protein, we have one-to-one complex, and from one-to-one complex, we have 2-to-1 complex. On the other hand, we can have dimer of the spike protein and then dimer by the ACE2 to also form one-to-one complex. Although these possible 2-to-1 complex have different conformation, using only mass or mass to charge ratio, we cannot really distinguish these conformation. So, however, there is multiple equilibria, like multiple interaction in here. So what I have shown you so far was for one-to-one interaction. How do we quantify a multiple equilibria? For that, the slow-mo was extended in the paper we published in 2024, where we developed, instead of taking the ratio of the abundant, a matrix of equation was developed to quantify for several relative response factor and hence the dissociation constant at the same time. And by using the extended slow-mo NMS approach, we could calculate the KD1 of ACE2 spike protein interaction of 0.5 micromolar, KD2 for the dimerization of the spike protein to be around 0.3 micromolar. So it can be seen that in solution, spike protein prefer to form the dimer than to form the complex ACE2. So caution should be taken when we're studying these interactions in solution. So I have showed you a different approach to study different protein glycan or protein interaction using native mass spectrometry. And it sounds like we have tools for everything, every type of biomolecular interaction we want to study. However, there's many other challenging systems that we want to look into. For example, if we have large heterogeneous protein interacting with the ligand, but when we do catch and release, no glycan is released or detected, then we cannot use a catch and release approach. We cannot even use a COI-NMS approach. And if we have unresolved spectrum, we cannot take the abundance and calculate the KD even with COI or slow-mo NMS. So with the development or the application of charge detection mass spectrometry, we are working on developing more method. Example is the studying of the interaction of chlorotoxin, the B subunit, which is a pentameric or protein interaction with the membrane platform. We call it the nanodisk. So the nanodisk itself is show a broad spectrum here. And the moment we add CDP5 in, we see the shift in the center of the spectrum. So we expect that there is some complex form. However, it is very challenging or impossible to say what are the complex, what are the stoichiometry. So by using charge index and mass spectrometry, again, we could resolve the interaction. So from the blob here, we could detect the free nanodisk 1 to 1, 1 to 2, and up to 1 to 6 nanodisk CDB5 complexes. And which is a bit strange to us, because if you look at the size of the nanodisk and the CDB5, we'll see that the size of the nanodisk, the diameter of the nanodisk is around 12 nanometer. The CDB5 itself is around 5 nanometer, and we expect to have 4 to 5 glycolipid would interact with the CDB5 on each side of the nanodisk. So the maximum stoichiometry of the complex that we expect was 2 to 1. So 1 CDB5 on each leaflet of the nanodisk. However, we saw up to 1 to 6. So the theory is probably in addition to the CDB5 binding to the nanodisk, there was the stacking of the CDB5, which hasn't been reported in the literature. So we search for collaboration. So Joe in the Young lab, Herbert with the negative stain TAM. So what we saw is with the lower resolution, we could saw something that looked like nanodisk with CDB5 stacking on top of each other. So because it's low resolution, ongoing work is to get better resolution of the complex and to confirm the stacking. However, with this observation, we proposed that from nanodisk, we have the CDB5 binding to both sides of the nanodisk, and from that, we have more CDB5 stuck on top of the other CDB5 to form up to 6 to 1 complex, the 1 to 6 complex. So in conclusion, I hope that I believe that we all know that glycans and protein-glycan interactions are very important in health and disease. And native mass spectrometry, from my point of view, or I hope that I have showed you, is a powerful technique to study protein-glycan interactions. And the choice of mass spectrometry tools really depends on the application, on the properties of the interaction that you want to study. So caution need to be taken when we use native mass spectrometry to study the interactions. And there's many other systems that is challenging to study with the currently available methods. So more development is needed. So with that, I would like to conclude my talk here today by thanking my village. So it takes a village to raise a child. And I'm very grateful for the support that I have been received from the Glycan group and the Mahal group, especially from Dr. John Glycan and Dr. Laura Mahal. And I would like to thank our collaborators. So here's collaborators whose projects are showed in the slide, and there's other collaborators as well. I would like to thank funding agency and special thank to Glyconet for the support for my time here and for the constant support. And what's next? So as Katrina already talked about, I'm moving to Waterloo in March to start my own group, and I'm happy to continue our collaboration, the existing collaboration, and expand my collaboration to study more relevant interactions in glycobiology, immunology, and thank you for the attention.
Wow, Dr. Bui, what an amazing talk. We do have some time for some questions if participants would like to unmute themselves and go ahead and ask. Perhaps I'll jump in then.
Okay.
I've heard quite a bit about mass spec and the different ways that you can use it, but I was curious which disease in particular you found the most enlightening when you were using mass spec to discover the interactions.
I have special interest in cancer. So yeah, so our ongoing projects right now is to look into the detailed mechanism of the recognition of the immune clearance or immune the escape of the cancer cells. So yeah.
Is that the work you're going to be doing at Waterloo?
Part of that, yes. So I already started here, so part of that is to continue that. The major part of my research in Waterloo is to look at the mechanism of the immune activations, and especially the innate immune activation, whereas the glycan are recognized by the lectins to activate the complement cascade. So yeah.
That's wonderful. Does anyone else have any other questions or comments?
I'm just going to add that if people have questions, because of the webinar feature is a little bit different, you can raise your hand and then we can unmute you if you have a question, or you could submit to the Q&A section.
Do we have any coming in there?
I don't see any questions. Oh, I see one, I think, in the Q&A. Do you have any other research topics?
Oh yeah, so for the my lab, we have 3 different directions for the research. So there's would be 2 broad direction with like subtopics in that. So we'll look at the biomarker discovery. We also want to look at the receptor discovery, which is very new, and then I'm very excited about that. And as I said, we'll look at the detailed mechanism of immune activation. Yeah, and then each of the direction have like several subtopic in that. And if you're interested in joining the group, we can talk further.
Next question. Did you find that the mixing rate changed depending on the concentration of sugars? Was this also correctable via your slow-mo method?
So yes, so the part of diffusion is it dependent of the concentration of the ligand. So the higher the concentration, the faster the mixing rate. And then because we try to model it, so it matters, but actually it doesn't matter much because we can fit for it. So we know, in addition to calculating the KD, we also fit for the change, the rate of the change of the concentration. And ideally, all of that should be corrected with the concentration independent mass spectrometry approach in theory. So, but in practice, usually I run a different concentration to see if the results are agreeable, and then a certain concentration when the change is too much. So the You can see that the gradient is too short, then we cannot reliably fit, then we usually need to dilute it more. So yes, it can be corrected, but we need to take it with caution. Like we need to run different concentration to see.
And another question, how small are the sample sizes that you work with in these cases? A lot of the time, we do not have a lot of protein or ligand, especially when we are working with big glycans, and this limits us from the detection techniques we can do. Would you recommend this type of detection when sample concentrations are very small over other techniques?
Yes, it's actually very helpful when you have very small amount of protein and glycan. So as a reference, the proteins that we use in here are usually at lower than 10 micromolar and sometimes like lower than 1 micromolar concentration. And the glycans are in micromolar range as well, so sometimes up to 1 millimolar. But usually we use very low volume, so the sample we usually receive is because there would be optimization and everything. So the protein that we usually start with is 50 to ideally 100 microgram, and the glycans can be lower than that. So it's like 10 to 100 microgram as well. And then for that, we can use for like several experiments. That is not one experiment. And yes, once you have that, you cannot really run MMR with that low amount and low concentration. So I think NMS is ideal for that situation. However, that is the ideal case. There is some very challenging system, like very challenging protein. Then we need more to optimize the method.
That's all the questions in the chat.
Okay, perfect. Please join me in thanking our wonderful speaker, Dr. Bui. She gave such an amazing talk. If you'd like to stay after and chat with her about her new lab, you're welcome to do so. Thank you so much for joining us here today. There will be an EDI survey that comes up shortly, and it will also be sent out with the copy of this video tomorrow. So please feel free to fill that out and send it back. And we hope that you join us at our next webinar, which is scheduled in February.
Do we have time for one more question? I just saw another one.
Okay.
Please go ahead.
Do you think with further development, this MS technique can be used for high throughput screening of synthetic glycans or glycomimetics for the drug discovery?
Actually, we can already do high throughput screening right now. So as you can see, depends on the molecular weight of glycan. We could accommodate more than 100 glycans at the same time. So it is very great for high throughput screening. For membrane mimetics, it is another problem because the molecular weight will be huge and will be different. So I think more development is needed, but my dream is like at some point, native mass spectrometry can do everything.
Thank you. Oh, another question just went in. Have you been able to distinguish glycan isomers?
Oh, that is another challenge in native mass spectrometry. So as you see, they in catch and release or in COI or in any mass spec data that I showed you so far, we distinguish glycan according to the molecular weight or the M over Z. To be able to distinguish isomer, we need another step. So either ion mobility that they have, can distinguish isomer, or we need to rely on the fragmentation of different isomer. So yeah, more development is needed in that field.
Thank you. I think that's all the questions now.
Perfect. Thank you. Yeah, we're thrilled to have you here today, and we really appreciate you being our first for this new year.
Yeah, thank you very much for the invitation again. I'm happy to be here today.
Thank you guys for attending and take care.
Okay, bye bye.